Hello everyone! Now we start studying the respiratory system, and these are our learning objectives. The respiratory system has several functions, and one of them is the one that we usually remember, that in goes the good air and out goes the bad air. So we have oxygen going into our body and we eliminate carbon dioxide. We need oxygen because of cellular respiration, and the oxygen is used to make ATP, and carbon dioxide is a waste product generated in cellular respiration as you make ATP. So you need the air reaching oxygen to get in and the air reaching carbon dioxide to get out. And this action of air going in and out of our lungs is what we call pulmonary ventilation. You need to think like it vents. And when you say something is venting, it's releasing air, right? So it's like moving air around. So pulmonary ventilation is related to the air coming in and out of our lungs. This function of the respiratory system is intimately linked to the cardiovascular system because we have the blood moving through our lungs to get oxygen, so we can use this oxygen to make ATP. Now, another function of our respiratory system is related to olfaction. So the fact that we can smell something is related to the respiratory system. And lastly, another main function of our respiratory system is related to vocalization. So the fact that we are able to communicate is also related to our respiratory system. We can divide the respiratory system in two sections, the upper respiratory system and the lower respiratory system. The upper respiratory system includes the nose, nasal cavity, nasal conchae, paranasal sinuses, and the nasal pharynx. So when we have an upper respiratory infection, the respiratory infection is in one or more of these areas, so it's closer to the surface of our body. Now, the lower respiratory system includes the larynx, trachea, bronchi, and then we have the bronchi dividing several times, and with each division the bronchi gets thinner and thinner and thinner until it divides into bronchioles. Now, looking at this diagram, we can see that our right lung is bigger than our left lung, and the reason for that is that our heart is shoved into the left side of our thoracic cage. Here we have a picture of a cadaver showing how the heart fits in between the lungs. Since the heart is slightly shifted to the left, we have one less lobe in our left lung. And the place in the left lung where the heart fits in is called cardiac notch. So we have two lobes in the left lung and three lobes in the right lung. And these lobes are separated by fissures. In the left lung, since we have just two lobes, we have just one fissure. We have the oblique fissure separating the superior and inferior lobes of our left lung. Now, our right lung has three lobes, the superior, the middle, and the inferior lobes. And separating the superior and the middle lobes, we have the horizontal fissure. And separating the inferior from the other two lobes, we have the oblique fissure. When we studied the heart, I mentioned the heart was located in the mediastinum, so the area in the middle of the sternum. Now, this medial surface of the lungs that face the heart is called mediastinal surface. The surface of our lungs that faces the rib cage is called costal surface, and the surface of the lungs that is in contact with the diaphragm is called diaphragm surface. Now, our lungs are like a spongy type of tissue, and if we could feel a lung, you'd notice that it feels like one of those fancy mattresses that when you press your hand, you leave an indentation, and then slowly the indentation is starting disappearing until the mattress looks flat again. So if you squeeze the lungs, it retracts, and then slowly it starts going back to the resting shape. And the opposite is also true. So if you stretch the lungs, it would recoil back to a resting shape. And that's because of the internal elasticity of our lungs. So if we think of that, we can ask ourselves what's keeping the lungs inflated, if it has a tendency to retract. And what keeps our lungs inflated is the pleura. Here we can see a cross-section of the thoracic cavity. And you can see that we have a space between the lungs and the thoracic wall. And this space is where we find the pleural cavity. We have the right pleural cavity surrounding the right lung, and we have the left pleural cavity surrounding the left lung. So if you recall when we talked about the heart, surrounding the heart we had the serous membrane of the pericardium, which was composed of the visceral pericardium and the parietal pericardium. And between those two layers we had the pericardium fluid. Similarly, here in the lungs we have the pleural membranes. We have the visceral pleural membrane right in contact with the surface of the lungs, and we have the parietal pleural membrane in contact with the thoracic wall. And between these two membranes we have the pleural fluid, which is a slippery serous fluid secreted by the simple squamous epithelial cells that make up the visceral and parietal layers of the pleura. So the parietal pleural membrane attached to the inner wall of our chest, and the visceral pleural membrane attached to the surface of our lungs. And then they prevent our lungs from collapsing. Now, the slippery fluid found in the pleural cavity between the parietal and visceral layers acts as a lubricant. And then it allows the two pleural membranes to slide against each other during respiratory movements. So, since the pleural membranes are what adhere the lungs to the inner thoracic wall, and with that our lungs are actually held to the wall of our thoracic cage, that's what keeps our lungs inflated. Now, if the pleura gets damaged, you can guess how bad that gets. When air enters either the right or left pleural cavity, so the space between the pleural membranes, air starts to flow into the chest, and the lung will pull away from the chest wall, and that results in either a partial or complete collapse of the lung. And this is clinically called pneumothorax, literally air in the thorax. Now, the way this is treated is by actually inserting the needle of a syringe into the pleural cavity, and then trying to aspirate the air into the syringe. And as the air is drawn back into the syringe and out of the pleural cavity, the lung reinflates and reattaches to the chest wall. So that's the importance of this pleural fluid in between the two pleural membranes. So in terms of functional anatomy, we can differentiate the respiratory system into the conducting zone and the respiratory zone. So the conducting zone, as the name implies, has the job of conducting the air until it reaches the respiratory zone, which is the place where the actual gas exchange happens. And as you can imagine, the place where the gas exchange happens needs to be very thin enough so the gases can just move across via diffusion. However, in the conducting zone, we have parts that are very exposed to the external environment. And as you go down into your lungs, it starts being less and less exposed to the outside environment. So with that in mind, you can expect a gradual change in the respiratory epithelium as you go from areas that are more externally exposed to areas really deep inside your lungs where the gas exchange happens. If we look at the air through a beam of light, we can see several little dust particles floating around, and we are actually inhaling all that. So we need to clean the air before it goes deep into our lungs. And in order to clean the air that we inhale, we have a ciliated mucous membrane lining basically the entire conducting zone of our respiratory tract, with the exception of the oropharynx and the laryngopharynx. The oropharynx and the laryngopharynx are lined with stratified squamous epithelium. So we have several layers, that's defined by the stratified word, right, of squamous cells. So oropharynx and laryngopharynx are lined with stratified squamous epithelium. But all the rest of our conducting zone, so the inside lining from our nasal cavity all the way down to the bronchioles, we find pseudostratified ciliated columnar epithelium with goblet cells. And this is the anatomical name for what I said before, ciliated mucous membrane. So since we are in an anatomy class, you guys need to know the anatomical name. So the name again is pseudostratified ciliated columnar epithelium. And to refresh our memory, when we say pseudostratified, it means that it looks like it has more than one layer, but the pseudo root means it's not true, right? So the pseudostratified epithelium looks like it has several layers, but in reality it's formed by a single layer of columnar cells, and in the apical surface of these cells we find cilia. So if we look here at this magnified version of the respiratory epithelium, we can see the goblet cells, which are labeled here as mucus cells, and the goblet cells are the ones responsible for secreting the mucus at which the particles and pathogens will get stuck to. And then we have the particles in the air that we inhale trapped in the mucus, and the cilia, all these cilia that we see right here, that are inside the mucus, they move in a specific direction, and with that they move the particles that were stuck in the mucus towards our throat, and we swallow it. And then all the dirty particles and possible pathogens that were present in the air we inhaled now go to our stomach, and they get destroyed by the acid of our stomach. So, as you can conclude, this mucus layer that we have inside our respiratory tract that traps all the inhaled dust particles and pathogens, it works as a respiratory defense system, right? So basically, throughout the entire conducting zone of our respiratory system, we have this mucus-ciliated membrane that traps the debris and pathogens that were present in the air we inhaled. But besides this cleaning function of the conducting zone also warms and humidifies the air we inhale. So if you put your hands in front of your mouth and you exhale, you can feel that the air you are exhaling is warm. And if you go in front of a mirror and you blow on it, you can see that the mirror gets foggy. And the only reason why the mirror gets foggy is because the moisture present in the air we exhale condensed on the mirror. So it's important to warm the air that we are inhaling because we are often exposed to atmospheric air that's much colder than our own body temperature. And for example, if we are in a very cold environment, we do not want that cold air getting all the way down into our lungs where it could potentially freeze our lungs. So we need to warm the air that we are inhaling. And also we need this air to get humidified, because humidification protects cells against dehydration. So if we inhaled really dry air that did not get humidified, it could dehydrate the membrane lining the inner side of our respiratory tract, and that could damage the cells of our respiratory tract. When we inhale air through our nose, the air passes through our nostrils, also called external nares. And then the air passes the nasal vestibule, which is this part at the beginning between the nostrils and the nasal cavity. The nasal vestibule is supported by hyaline cartilage that we have at the tip of our nose, and that's why the tip of our nose is kind of bendable. At the nasal vestibule, we find hairs that extend towards our nostrils, and these hairs found here at the nasal vestibule work as strainers, inhibiting large particles from getting into our nasal cavity. Now, after the air passes through the nasal vestibule, the air reaches the nasal cavity. And we have the right and left nasal cavities. And the wall we have separating the right and left sides of our nasal cavity is called nasal septum. And it is formed by the perpendicular plate of the ethmoid bone fused to the vomer bone. And forming the anterior portion of our nasal septum, we have hyaline cartilage. And that's what confers the flexibility of our nasal septum. When air reaches the nasal cavity, the air encounters some speed bumps, which are the nasal conchae. We have the superior, middle and inferior nasal conchae working as speed bumps, creating turbulence in the air we just inhaled. And this turbulence makes the air move around, and with that, dust particles and pathogens that were present in the air we inhaled get stuck in the mucus. And the air gets cleaned, warmed, and humidified before it follows its path to our lungs. So if we did not have this bumpy surface, if instead we had a very smooth surface, the air we would inhale would just flow very nicely through the nasal cavity and follow the pathway down to our lungs, and that would not be very helpful. So the presence of these elevations obligate the air to move around, and this moving motion warms and humidifies the air and help with the dust particles and pathogens getting stuck to the mucus. Now, besides this turbulence created by the nasal conchae to help us clean, warm, and humidify the air, this turbulence also helps us to smell better. So, due to the turbulence, we have odorant molecules getting trapped in the mucus, and with that, these chemical molecules end up binding the olfactory receptors present at the olfactory epithelium on the superior surface of our nasal cavity. Hence, the turbulence created by the nasal conchae aids in the process of smell. Now, after the air swirls around in the nasal cavity, it keeps moving backwards, and after the air passes an area called posterior nasal aperture, the air reaches the pharynx. And the pharynx is shared by the digestive and respiratory systems. And it is divided into three sections: the nasal pharynx, the oral pharynx, and the laryngeal pharynx. The portion of the pharynx that's close to our nose is called nasal pharynx. The portion of the pharynx that's close to our oral cavity is named oral pharynx, and the portion that's closer to our larynx is called laryngeal pharynx. So it all makes sense. Now, right at the back of our nose, as I just said, we have the nasal pharynx. And if you recall when we talked about our ear, I mentioned that we had the auditory tube, and this tube was responsible for equalizing the pressure on both sides of the tympanic membrane. And this tube opened up in the nasal pharynx, and here you see it. This opening of the auditory tube receives the name of pharyngeal opening of the auditory tube because it opens in the pharynx. Now, if you look here, separating our nasal and oral cavities, we have these two structures, the hard palate followed by the soft palate. And the hard palate is made by the maxilla and the palatine bones, and the soft palate is soft, and it moves up and closes our nasal passage when we are swallowing. The soft palate is what designates the line that separates the nasal pharynx and the oral pharynx. And a very interesting thing is that the epithelium changes when it crosses from the nasal pharynx to the oral pharynx. In the nasal pharynx, we have the regular respiratory epithelium, which is the sealed stratified ciliated columnar epithelium. But now in the oral pharynx and also in the laryngeal pharynx, which is below the oral pharynx, we have stratified squamous epithelium. So several layers of squamous cells. And if you pay attention to where the oral pharynx and the laryngeal pharynx are located, you can see that these two parts of our pharynx are on the way of the food that we swallow. So in this area, we need several layers of cells, so stratified squamous epithelium, to ensure that the oral pharynx and the laryngeal pharynx can resist to all abrasion caused by the food we swallow. Now, as I just mentioned, the laryngeal pharynx is below the oral pharynx, and the hyoid bone that we see here is what marks the line that separates the oral and the laryngeal pharynx. So the laryngeal pharynx starts at the level of the hyoid bone, and it goes until the entrance of the esophagus. And as you can see, the laryngeal pharynx is right behind the larynx that we see here. Now, the larynx is our voice box, and this is the place where we find our vocal cords. Three very noticeable pieces of cartilage make up our larynx: the epiglottis, the thyroid cartilage, and the cricoid cartilage. The thyroid cartilage is this big piece of cartilage that we see here, and this is the place where we find the laryngeal prominence, which is commonly known as Adam's apple. Right behind the laryngeal prominence, we find our vocal cords. In other words, the laryngeal prominence is the place where our vocal cords are anchored anteriorly. As you know, men have a more prominent, bigger Adam's apple than women. And due to testosterone, the larynx of a man is bigger than the larynx of a woman. And since we have the vocal cords in the larynx, that results in the voice of a man being deeper than the voice of a woman. Now, below the thyroid cartilage, we find the cricoid cartilage. And you can see here in the posterior view that the cricoid cartilage is the only cartilage of our larynx that has a posterior surface. All other cartilages of our larynx do not have a posterior surface. Now, between the thyroid and the cricoid cartilage, we find the Cricothyroid Ligament. And this ligament is clinically relevant because this is the place where tracheostomy is done. Because at this position, you know you would not damage the vocal cords of the person that's being unable to breathe. So you make a little hole in the cricothyroid ligament and you allow the air to go into the person's lung in case the obstruction of the airway was located above this area. Here we can see the glottis, which is this opening we have in our larynx. And in the glottis we find the vocal cords. That's why we say that the glottis is our vocal cords per se. We have true vocal cords, which are called vocal fold, and we have false vocal cords, which are named vestibular fold. And as you can see, the vestibular folds are located on the outside of the vocal folds. They are called false vocal cords because they are not elastic enough to vibrate when air passes through them, so they cannot produce sound. So what happens is that when we breathe in, the glottis opens up and air goes into our lungs. Now, whenever we talk, we are pushing air out of our respiratory tract, and when the air passes through our glottis, it vibrates the highly elastic vocal folds and creates sound waves. In fact, if you put your hand on top of your voice box, which is your larynx, you can feel it vibrating, and that vibration is what is producing the sound. So as you can imagine, depending on the tension, length, and diameter of the vocal folds, different sounds can be produced. And if we bring the vocal folds very close together, we can produce very high-pitched sound. And if we put them apart, we can produce very deep sounds. And in order to tense or relax these vocal folds, we must have muscles in this area. And these are called the intrinsic laryngeal muscles. So the intrinsic laryngeal muscles regulate the tension in the vocal cords, allowing us to produce deep sounds or high-pitched sounds. And also these intrinsic laryngeal muscles, they control the opening and closing of our glottis. So if we have intrinsic laryngeal muscles, we also have extrinsic laryngeal muscles. Because if we did not have both, you would just say laryngeal muscles, right? So the extrinsic laryngeal muscles, they work extrinsically and they connect the larynx to the nearby structures. And together with the movement of the hyoid bone when we swallow, the extrinsic muscles in our larynx move up and down. Now, you can ask me, why do we have the false vocal cords if they seem to not do much, right? So the vestibular folds, which are the false vocal cords, they are mostly involved in helping to close the glottis. So during the Valsalva maneuver, which is the name given to the action of us forcing exhalation with our airways closed, we have the vestibular folds keeping the glottis closed and preventing the air from escaping. And with that, we have the increased pressure in the abdominal area, and that helps during lifting of heavy weights and also during defecation, urination, and during the delivery of a baby through the vaginal canal. Here we have an anterior view and a sagittal section of our larynx. And one of the cartilage pieces that form our larynx is the epiglottis. Looking at the structure of our larynx, we see that the hyoid bone supports our larynx superiorly. And connecting to the hyoid bone, we have several swallowing muscles. When we swallow, these muscles pull the hyoid bone, and consequently the hyoid bone pulls the larynx up. And when that happens, we have the epiglottis covering the glottis. And with that, the food we are swallowing goes into our esophagus and not into our trachea. So if you put your hand in your neck area and you swallow, you can feel the larynx going up. And when the larynx is up, we have the epiglottis closing the entrance of our glottis. Now, under the cricoid cartilage of the larynx, we have the trachea. And the trachea is this long tube that starts below the larynx and finishes with the split of the right and left bronchi. All this cartilage that we see here, the cartilage making up the thyroid and the cricoid cartilage of the larynx, the cartilage making up the trachea, and the cartilage making up the bronchi. All this cartilage is hyaline cartilage, which is the type of cartilage that's tough, but it is kind of flexible. And that's why if we press our larynx, we can feel it moving. So the hyaline cartilage, as all cartilage, is avascular, but compared to the other types of cartilages, the hyaline cartilage is the weakest type. And we find the hyaline cartilage between the ribs and the sternum, in articular cartilages within synovial joints, and in the passageways of our respiratory tract, as you can see here. Now, going back to the trachea is a tough, flexible tube that's held open by C-shaped hyaline cartilage rings. And if you take a look at this cross-section of the trachea, you can see that right behind the trachea, we find the esophagus. So when you feel the front of your neck, that's the trachea, and the esophagus is right behind it. So the esophagus is posterior to the trachea. Lining the lumen of the trachea, we find the regular respiratory epithelium, which is the sealed stratified ciliated columnar epithelium. Now, look here at the ends of the C-shaped hyaline cartilage rings of our trachea. You see that connecting the two ends, we have a smooth muscle named trachealis. And as all smooth muscles throughout our body, the trachealis muscle is under involuntary control. And as you can suspect, based on its location, if the trachealis muscle contracts, it decreases the lumen of the trachea because it brings the endings of the C-shaped ring together. And if the trachealis muscle relaxes, the trachea dilates and the lumen of the trachea increases. Now, you might be wondering why this hyaline cartilage is C-shaped instead of a full ring. And the reason for that is because the esophagus is right behind the trachea. And through the esophagus, we have the food we swallow passing through. Now, if the ring was a complete circle, the swallowed food would go down our esophagus, bumping into every single ring, and this would cause no pleasant sensation. It would be very unpleasant. So, the C-shaped cartilage we have in our trachea allows the swallowed food to go down our esophagus smoothly. And in case we swallow something too big, we have the trachealis muscle getting a little pushed into the trachea lumen, and with that, the food keeps going down until it reaches our stomach. Now, after the air passes through the larynx, it flows down through the trachea, and then it reaches this split point where we have the carina of the trachea. The carina of the trachea is very important because we have several nerve endings grouping together at the carina. So, in case the breeze are capable of getting into our trachea and get all the way down here, reaching the carina, we have the free nerve endings at the carina getting stimulated, and we start coughing like crazy. So the stimulation of the nerve endings at the carina leads to the coughing reflex. And the coughing reflex is a defense mechanism because by coughing, we can remove the breeze out of our airways. Now, when our trachea is split, we have the right and left main bronchi. And if we compare these two main bronchi, we can see that the right main bronchi is much wider and also kind of steeper than our left main bronchi. So if you think that we have 3 lobes in the right lung and just 2 lobes in the left lung, it makes sense that our right main bronchi is wider than our left main bronchi. Now, taking into consideration that our right main bronchi is much wider and steeper than our left main bronchi, if an object goes down our trachea, would you expect the object end up in the right or left lungs? The answer is right lung, right? Because the right main bronchi is wider and steeper than the left main bronchi. So clinically, what's found is that if someone aspirates a foreign object, it is most likely to end up lodged in the right lung. And interestingly, the cigarette smoke is more likely to go into the right lung than in the left lung. And so when we look at the smoking-related lung cancer's incidence, the data reveals a higher incidence on the right lung than on the left lung. But this higher incidence could also be related to the fact of us having 1 extra lobe in the right lung than in the left lung. Each main bronchi enters the mediastinal surface of our lungs through the hilum. And actually, the hilum is the site of entrance and exit of all structures associated with our lungs, including our blood vessels. Now, after the main bronchi enters the lung, it starts splitting into smaller and smaller branches. And this system of tubes that we see here that's formed by the main bronchi and their branches is called the bronchial tree. And we can subdivide our lung lobes into bronchopulmonary segments. And each bronchopulmonary segment is supplied by a tertiary bronchus. Now, you don't need to worry about all these segments because I will not be testing you on that. After the main bronchi enters the lung, it subdivides into secondary bronchi that subdivides into tertiary bronchi, and it keeps subdividing like 24 more times until we get to what we call bronchioles. Now, this little bronchiole splits more until we get to alveolar ducts, and at the end of the alveolar ducts, we have the alveolar sacs, also called alveoli. As you can see, there are some changes in the histology of the wall of these airways as we get to smaller and smaller divisions. So you can observe that with each subdivision, as we go from larger to smaller and smaller airways, we progressively have less and less hyaline cartilage. So if you remember our trachea, we had the hyaline cartilage in the wall, and that was what prevented the trachea from collapsing. But as we go into smaller and smaller and smaller airways, there is progressively less hyaline cartilage in the walls, and therefore the smaller airways are capable of collapsing. Now, another progressive change that is observed is that when we go into smaller and smaller airways, there is an increase in the amount of elastic connective tissue and also in the amount of smooth muscle. So the very small airways are very flexible and have a thick layer of visceral smooth muscle in the wall, and this smooth muscle is innervated by autonomic motor neurons that can cause the visceral smooth muscle to constrict, so it narrows the airway, or to relax, and in that case we have the airway dilating. Here we have a magnified view of the alveolar sac. And we can see that reaching the alveolar sac, we have the alveolar duct. And each lung has about 150 million alveolar sacs. And that's what confers our lungs the spongy texture it has. All these alveolar sacs are surrounded by capillaries. And as you know, the capillary wall is formed by simple squamous epithelium, which is the same tissue that forms the walls of these alveolar sacs. So this area right here favors the gas exchange, and in fact this is the only area where exchange can happen because the walls of both structures are thin enough to allow gases to cross. So we have oxygen that is inside the alveolar sacs getting out and going into the blood, and the carbon dioxide that is inside the blood getting out and going into the alveolar sacs. As you can see, we have bands of elastic fibers surrounding the alveolar sacs, and this elastic tissue helps in the maintenance of the shape and position of each alveolar sac during inhale and exhale, because as I mentioned before, we do not have cartilage in this area, because the presence of cartilage would not allow the gas exchange to happen. Now, I just mentioned the walls of the alveolar sacs are made of simple squamous epithelium, so a single layer of squamous cells, but these cells receive the name of type 1 alveolar cells. Now, scattered among the type 1 alveolar cells, we have another type of cell called type 2 alveolar cells. And the type 2 alveolar cells are responsible for secreting surfactant. If you recall, the air that we inhale gets humidified. And what happens is that when this humid air gets into these little sacs, and then we exhale, we remove the air out of these little sacs, the humidity that was present in the air would make the walls of these little sacs to glue to each other, leading to the collapsing of the alveolar sacs. And consequently, gas exchange cannot happen. So this surfactant produced by the type 2 alveolar cells work as a detergent, and the presence of this detergent does not allow the sides of the alveolar sacs to glue to each other, because the detergent reduces surface tension, and then you can keep breathing in and out. We also find inside of the alveolar sacs macrophages. And the function of these alveolar macrophages is to engulf any virus, bacteria, or debris that are capable of getting into our lungs. And with that, the macrophages help us to not get sick. Now I would like to share with you some diseases related to the respiratory system. So here on the left side, we have the alveolar sacs of a healthy person. And on the right side, the alveolar sacs of a person with emphysema. Emphysema is associated with the chronic progressive degeneration of the lungs. And in a person with emphysema, the alveolar sacs degenerate. And this is chronic, which means that it doesn't go away and it does not get better over time. And by looking at this diagram, you can see that emphysema not only affects the airflow, but it also affects blood flow. And there is a dramatic impact on the ability of the oxygen to get to the alveolar sacs, and also for the blood to flow through the capillaries of the alveolar sacs to receive oxygen. You probably have seen a person with emphysema, and they are usually too weak to walk, so they drive around in those little electric carts, and they are usually carrying their air tank with them. Tobacco smoke is by far the most dangerous behavior that causes people to develop emphysema, but emphysema is also associated with exposure to air pollution and chemical fumes and dust. In a very small percentage, some very rare cases, we have emphysema caused by an inherited deficiency of a protein that protects the elastic structures of the lungs. Now, most people with emphysema also have chronic bronchitis. And bronchitis can be acute or chronic. And the chronic bronchitis is the one that does not go away and it doesn't get better over time. Bronchitis is an inflammation of the lining of the bronchial tubes, which are the tubes that carry air to and from our lungs. So people who have bronchitis, they often cough up thickened mucus, and they try to eliminate that mucus from their airways. Now, asthma is a disorder characterized by the conducting airways contracting too much and too easily. So it is related to the tightening of the smooth muscles that surrounds our airways. And an asthmatic attack can happen spontaneously or after the exposure to a wide range of stimuli. During an asthmatic attack, there is inflammation in the air passages, and that results in a temporary narrowing of the airways that carry oxygen to the lungs. And this leads to shortness of breath and chest tightening. And if the asthmatic attack is very severe, it even decreases the ability of the person to talk. The last thing we need to talk about is the respiratory muscles. And the important breathing muscles are the diaphragm, which separates our abdominal and thoracic cavities, and the intercostal muscles, which are the muscles between our ribs. And intercostal literally means between the ribs. In order for us to inhale, we have the respiratory muscles contracting. Now, when these muscles contract, they increase the chest volume, and that creates a suction that draws air into our lungs. As you know, our lungs rest on top of the diaphragm, and when the diaphragm muscle is relaxed, it arcs up, and it looks like this. Now, the diaphragm muscle is connected to the wall of our abdominal cavity, so all the sides of our abdominal cavity, because it separates the thoracic and abdominal cavities. And when the diaphragm muscle contracts, it gets shorter. And as you know, the shortest distance between 2 points is a straight line. So when the diaphragm muscle contracts and it gets shorter, it becomes a straight line. And with that, the chest gets bigger from top to bottom. And when the intercostal muscles contract, that enlarges the rib cage. So it increases the chest from side to side and from front to back. So the entire chest volume increases when these muscles contract, and that lowers the pressure inside the chest and lungs, and then we have the air getting sucked into our lungs. And if you put your hands in front of your nose and you breathe in, you can feel the air getting sucked into your respiratory tract. And if you inhale very deeply, you can literally see your rib cage expanding. Now, when the diaphragm and the intercostal muscles relax, the chest volume gets smaller. And if the chest is smaller, the pressure inside the chest increases. So the air is forced out of our lungs. So, as a summary, when the respiratory muscles contract, we inhale. And when the respiratory muscles relax, we exhale. And with this, we finish this chapter. Please let me know if you have any questions. Bye!